NBG.
If my internet plays this game with me, I'm gonna. I don't know what I'll do. I'll do something.
We'll keep it going.
Got rid of CenturyLink, so this wouldn't happen.
Yeah, well,
it's happening. It's happening, and we're live.
When will then be now? Soon.
Uh-oh.
What are you owing?
Is it? Can you still hear me?
Okay. Oh yeah, you sound beautiful.
I don't know because y'all froze up and then I can still hear you.
No, we're good, man. We're good. We're live. We're 40 seconds in.
Okay.
Sure Snakes and Stogies.
This is Snakes and Stogies episode 134. Is there a lag?
No.
Okay.
I think there is a lag on your end.
Carry on the intro.
Is there a lag?
I don't know. I can't tell.
I don't think there is. Do you want to keep going? You were doing such a good job.
Yeah. Yeah, this Snakes Stogies episode 134. My name's Justin. I got a problem with my internet. This episode is brought to you by BlackBoxCages.com. Check them out. Facebook, Instagram. Right. Nah, I can't.
Center your screen, people.
Right there. Check them out. Like I said, Facebook, Instagram, BlackBoxCages.com. That's right. Right up top. Get you a rack, get you a cage, treat yourself. And then Puget Sound Pythons, the good, fine folks of Jeff and Kendra up in the Pacific Northwest. Give them a follow, Facebook, Instagram as well. They have some stuff, I believe, that will be being posted for sale soon. I don't know exactly when or what, but they've been busy with some neonate action. So check them out. And yeah.
Rock and roll.
We are here this week to talk about some snake fungal disease. We have Alex Romer with us. He was actually one of the first guests on THP a couple of years ago. So we've been in communication here and there since. And he hit me up a while back, actually, about doing a fungal disease episode. And I figured that's something that really you don't hear about as much anymore. I know at one point it was in the headlines a good bit. On social media and news outlets and things like that, but it seems like it's kind of disappeared from the media circle. So, figure we talk about it a little bit, see sort of what's new, any updates there. I believe we're going to be joined by Dr. Donald Walker here shortly, who is also working with Alex on studying this and.
Good stuff, man.
Am I still lagging?
Good stuff. Yeah, you're lagging a little bit. You sound good, though. Don't worry about it. Keep going.
I must be.
Oh, yeah.
Snake cheese.
Definitely lagging. Yes. Billy Jenkins, always painting a vivid picture.
What are you smoking tonight?
Tonight, I had a customer come by my shop, and he was so happy that with his stuff that he gave me a 601 La Bomba. So... I don't even know what size this is. It's a really, really long son of a gun, but that's what we're driving.
A Lonsdale.
What are you smoking?
Romacraft Cro-Magnon.
Nice.
Oro.
You're a little fuzzy, but we could see that band.
I'm thinking it has to do something with StreamYard because we have not had this issue at all. And now all of a sudden it's struggling. And
Are you sure it's not, Cassidy Smith?
I'm fairly certain it's not Cassidy Smith because nothing's changed.
Okay.
So I don't know. We'll roll with it and see what happens.
As long as Alex and I look as good as we do right now, that's all that matters.
Yeah, the difference between this and Saturday night when me and Jake were doing this live is like you're not here, so you can if I'm in and out constantly, you can at least
carry
it.
Yeah. And right now you're crystal clear both visually and audio, so yeah. Alex, are you imbibing in any adult vices this evening?
I'm sitting in my office at work, actually. It's where I thought I would get the best internet connection, so unfortunately not.
That is no excuse at all. You're off the clock, man. You don't have like a fifth of bourbon in the desk drawer or something? Isn't that what all you science guys have?
No, I'm not that cool, unfortunately.
You could be with that mustache. You could be.
Maybe once I get tenure, you know.
Nice. Yeah. There's just a little snifter of gin in that keyboard drawer. Oh man. Well, again, welcome back to the show, man. It's good stuff.
Thank you.
And let's do this. Smitty, as you light that cigar, how's babies? How's those Texas beauties that you got?
They're large and in charge and angry. And I am very much looking forward to getting them out of my house. They're pretty snakes, but I'm officially sort of at the point where I'm like, it's kind of like candy corn. I can have it once a year and be good. That's where I'm at with thorn scrubs. So,
yeah,
hatched out the 5. They're cool. Of course, Chris doesn't want any of them. He's like, you son of a bitch.
Why would he?
I don't know.
He pawned the parents off on you.
He did. Well, actually, the parents are getting shipped off later this week. So,
they're that pleased with them.
Yep. Gonna get babies eaten, which I don't see being an issue. I mean, they could be eating fuzzies like straight out of the egg. They're huge, gigantic. Other than that, I mean, they're just angry little thorn scrubs. So
that'll
happen.
Thing. Hello.
Was ist los, Herr Doctor? Oh no, is he lagging now?
Why you got to make it weird, Phil?
I just met the guy. I got to be weird.
I'm lagging.
Patrick said StreamYard has been funky on aquarium live streams lately, so it is likely a StreamYard problem.
Okay, fair enough.
If it ain't broke, don't fix it. It was doing so good for so long, and they decided to start tinkering with it.
I know they gotta like update things.
Y'all there? Can you hear me?
Yep.
Yes.
Yes, sir.
Okay. How you doing?
Us?
I can. Really sorry I got the time zone wrong.
Oh.
Quite all right. Well, welcome to the show.
Thank you for having me.
So, do you guys want to just jump right into this, or do you want to do some? Why don't we do some introductions first? Whichever one of you guys wants to go first, give a little bit about yourself and refresh the listeners, and kind of give a little history of how you got where you are.
Well, Donnie's the professor. I feel like he should go first.
Oh, sure. Yeah. So, my name's Donnie Walker. I completed my PhD at Rutgers University. Actually did my undergrad at the same university that Alex went to, SUNY ESF. I'm traditionally trained as a mycologist, but I've started to work in microbial ecology for probably maybe the last 10 years or so. I've been studying the snake fungal disease system for approximately 7 to 8 years now.
Wow, that's awesome. Very cool. So are you a herper by nature, or did you kind of become one because of the fungi?
Yeah, kind of I met some really interesting people along the way that convinced me to study herps, that I should run and chase after them in the field and study the microbiomes and the different fungi, bacteria that live on their skin and in their gut. So I am not traditionally trained as a herpetologist. In fact, I've never actually had a herpetology course before. But I love to chase after them. It's a good time.
That's great, man. The field work is addictive when you're captivated. You know what I mean? It
sure is.
You've got when you're working with awesome animals, I imagine it just gets better and better. And even if you're not that into the animals themselves, it's still the hunt of it that I think is a lot of fun on its own. And you're hunting for multiple things. You're hunting for the fungi and for the animals. So like, it's awesome.
It's so true. Yeah. To be honest, I mean, I do love our hosts. Love to flip logs and catch cool salamanders and chase after frogs in ponds and the occasional snake capture. I mainly let Alex do that type of work, but yeah, I enjoy it definitely.
Yeah. How Alex? How do you tie into the thing? How would you get involved in the SFD stuff?
Yeah. So it's kind of funny that we're talking now, because when you guys first spoke to me, it was like right when I had moved to Tennessee to start my master's work, which is when I also had just started working with Donnie. And so I worked with Donnie for 2 years to get my master's degree, studying snake fungal disease. And then I kind of bumped around for a little bit after I got my master's. I worked as a technician at a couple of positions where they were doing research on cancer. I worked as an intern at the Chattanooga Zoo for a semester. And then ultimately Donnie got back to me and was like, would you like to come back to work for the lab as a lab manager? And so that's what I'm doing now. And so I do still work on snake fungal disease, but we work on a couple other projects. So we're also working on a fungus that lives widely in the gut of amphibians, reptiles that eat insects, called Basidiobulus. And so that's when Donnie talks about us being in the field and capturing, frogs and salamanders and snakes and that kind of thing. That's something that we do as part of that project. We do still opportunistically collect samples for snake fungal disease and things like that. So we do our lab in general works on that interface of understanding the microbiology of reptiles and amphibians for most of our projects.
Yeah, Alex just got out of the field probably about, I'm guessing 20, 30 minutes ago.
About an hour, just enough time to catch a shower and look presentable for this podcast.
Nice. Did you catch any wood frogs?
No, just released the ones I had.
Okay.
Yeah.
Super cool, guys. Super cool.
Yeah. So snake fungal disease, that was something that I remember reading a lot about at least a couple of years ago. Inside of the last 3 or 5, I think, is when it was there was, I guess, a borderline sort of panic about it. Whether that was sort of just an initial thing or if it is still very much so a problem, I don't know. But we can sort of get into, I guess, what it is. Is it a single fungus? Is it sort of a family, in comparison to kind of like the Nido stuff? I know they're completely different, but is it more of like a group of fungi that are doing it? Is it just a single one? Sort of what's the explanation of what it is and what it does and why it's such a problem?
Yeah, I think that's a really good question. So the term snake fungal disease kind of comes from the hazy origins of the origin of that disease. So it was around 2008, 2011 that snake fungal disease, or a fungal disease, started to be documented in pit vipers, mostly in the northern United States. So Denver rattlesnakes in the northeastern United States, Massasaugas, more in the Midwestern United States. And it was unclear what was causing these, either dermatitis or lesions or facial swelling. And so it wasn't known, was this being caused by one pathogen? Was this being caused by multiple pathogens? But it was understood that it was being caused by a fungal pathogen. So that's why it was assigned the term snake fungal disease, because that was basically the finest ecological agent that could be assigned to the disease at that time. And since then, there's been some taxonomic reassignments of the pathogen that causes snake fungal disease. But we now know that it's caused by a fungus, Aphidiomyces aphidicola, and it's caused by a single pathogen, that organism. And so there's recently been a push to refer to it as aphidiomycosis in place of snake fungal disease, as a result of the fact that we know it's caused by this particular fungus, as opposed to kind of just a broader fungal category. And whether it should be renamed or not, I think there's a debate to be had there, because people generally know this disease as snake fungal disease, whereas aphidiomycosis is a more specific term, but perhaps disconnects people from some of the earlier documentations of the disease.
Yeah, it's been thought of as and called hibernation blisters or hibernation sores quite often, I think, by herpetologists of the past before Ophidiomyces was named and documented. So herpetologists used to document this when snakes would come out of hibernation, and there'd be facial lesions, large granulomas, just necrotic skin, just odd looking disconfigurations on the animal.
Yeah, Phil, can you pull up a picture of it? I'm afraid that if I pull up a picture and ask this thing to do anything other than what I'm doing, it might explode and kick me out.
I actually was going to try to, and then my screen started lagging, and I was like, "Oh no, let me try and pull something up."
Yeah, it's pretty brutal, pretty gruesome. Oftentimes happens on the head and the eyes of the animal. What people think, not totally certain that it causes death, but perhaps could result in issues with the animal being able to see and sense predators. Oftentimes it's thought that the animals lay out and bask in the sun to attempt to elicit a febrile response and suppress the pathogen. Yeah, there you go.
Yeah, it's pretty gnarly.
Yeah, it's pretty gnarly, definitely. And when they can't see, then unfortunately they succumb to predation.
And I would honestly say a picture like that is probably more typical of what you see on most snakes in the field. So like, even though like something like what you have the cursor over right now is like where you'll see that on headlines where it's an animal that like is extremely disfigured.
Yeah, it's got like the elephant man thing going on.
Right. It's very rare to see an animal like that in the wild. It's much more common to see perhaps something like that, or perhaps even a little less severe than that on most animals.
To be honest, I think. Oh, go ahead, Alex.
Yeah, I was just going to say most studies that look at animals through time find that there isn't significant mortality in the field associated with snake fungal disease. And there are studies. So, for example, in one of the initial inoculation trials with the Ophidiomyces, where they attempted to determine is this the actual fungus causing snake fungal disease, I think they had a 40% mortality rate, but that was also with 5 snakes. And so there was a concern that it could be causing high levels of mortality. But when you actually track animals in the landscape through time, it doesn't necessarily seem to be killing animals outright. And often animals that do have severe lesions when they're followed through time, their infections kind of clear over time and perhaps do also get more severe over time. But it's not like a linear progression to death or severe disease like it might be is presumed if you like look at pictures online.
Yeah, I mean, the way the news worded it made it sound like it was like the chytrid of snakes, where it was like anything that gets it is toast. You know, it's taking over everything. Everything's dying. Like they really, I mean, it's the news, so I kind of expect it, but they really made it seem like it was the end of times for a lot of species.
Yeah, to be honest, I think we just don't quite know. This fungus has really only been studied in detail for probably the last 10 to 12 years. So not trying to downplay it, it could tentatively be problematic, but I think it's thought that it's oftentimes like opportunistic, like secondary infection by maybe different bacteria or alternatively fungi that cause these snakes to die or succumb to predation or something like that. Yeah, to be honest, like we've sampled like upwards of 1,000 snakes in the field now, and I've only really seen really bad snake fungal disease, like those nasty pictures that you're showing, maybe 2 or 3 times. I think these animals are probably getting picked off by predators long before they look real bad like that.
Does it have any effect on sort of their what's the damn word? Metabolism? Like, does it snakes that have it, are they more lethargic? Is there any difference in terms of that, or is it literally just like a physical, like I can't see what's going on kind of thing?
So Craig, Lind, and Aguilero, I believe, is the other investigator from Florida, do work on pygmy rattlesnakes there. And so they've done a lot of work on sublethal consequences of snake fungal disease in those populations. And so one study that in particular that comes to mind is they found that infection with snake fungal disease was associated with increased basal metabolic rates. So just having snake fungal disease, those animals had higher just resting metabolism. And also they had increased water loss compared to animals that didn't have snake fungal disease. They also found that snakes that had snake fungal disease, males during the end of the summer, spermatogenesis, when they're getting ready to breed, and during the actual breeding season, had lower testosterone than males that didn't have snake fungal disease. And females had lower levels of the pheromone or hormone that stimulates vitellogenesis during spring, when they should be producing embryos. So even though snake fungal disease might not be killing snakes outright in all cases, it definitely has sublethal consequences that are probably relevant to the host.
Yeah, and two of the somewhat recent studies that were published that actually fulfilled Koch's postulates to basically show like the fungus does cause this disease and does result in some lethal effects to snakes. They found that there were issues with like righting reflex, and the snakes were like laying out in the open more often than they typically would be under cover objects, and a variety of other behavioral issues that they documented.
Do you guys feel that that's some kind of association with them attempting to heal themselves by, getting sunlight or raising body temperature?
UV.
Yeah.
Yeah, Alex, you can talk about some of the modeling that you did with Cody's work.
Yeah. So there's there's something called the febrile response in reptiles. You guys might already be familiar with it, but it's the idea that
I'm not.
Oh, okay. That reptiles will raise their body temperature using their external environment as opposed to like endothermic mechanisms when they're sick to fight disease more effectively. So it's kind of
like how we have a fever.
Exactly. But instead of doing it with their metabolism, they do it by like maybe basking at a higher temperature or something like that. And so that was, for example, when snake fungal disease was still relatively new, they followed a couple of rattlesnakes and timber rattlesnakes in the Northeast that had this fungal dermatitis, and found that they were basking out in the open later in the year. At times it seemed inappropriate. And so there have been these anecdotal reports that like snakes with snake fungal disease tend to bask in like later times of the year, times that seem inappropriate. And so perhaps they're attempting this febrile response. And our lab about 2 years ago, I think at this point, with the Cody Godwin and Chris Murray, who's now at Louisiana State University, had water snake or ribbon snakes shipped to us from a wholesaler, and we were going to inoculate them and then put them on a thermal gradient so that way we could see did snake fungal disease have an effect on their preferred basking temperature. But actually very relevant to this podcast. So this comes from an animal wholesaler. These animals could have potentially gone into the hobby, but when we got them, they had snake fungal disease. And so that kind of put a wrench into our experiment. And so ultimately we wound up looking at kind of different categories of disease. So for example, like animals that already had disease and we inoculated versus animals that had never had disease that we inoculated. So like a naive versus the conditioned immune response and things like that. And the biggest difference that we saw in the gradient was that those animals that had already had disease and then we gave them a second dose of disease found a more stable, they found a substrate temperature that they wanted faster, and they were more stable at like, this is the temperature that I want to be on, than those animals that had never seen disease before. Whereas we didn't necessarily find a difference between those animals that weren't inoculated and those that were inoculated. So, it's worth, there haven't been a ton of studies on that particular subject. It would be interesting to see more people do that experiment and see what results they get. But I think there is definitely anecdotal evidence and some experimental evidence to suggest that snake fungal disease does have effects on how snakes are basking.
It was a bit unexpected.
Yeah.
But yeah, typical in the sciences.
Everything about it was unexpected, including when we got a bag full of snakes and half of them had snake fungal disease.
Now, is this something that is regional, where, for example, the snakes that you got from the wholesaler may have been South American, and obviously timbers are native to North America? Is it something that may have gotten loose into the local population, or is it something that's just from crappy conditions and bad husbandry?
Yeah, that's a great point. So these snakes were collected in Florida because they were ribbon snakes, but in terms of where the actual where they collected disease from, my guess is that it would be from snake fungal disease that is found in that area of the country. There was a recent paper that showed that there are different clades of snake fungal disease. So basically, depending on where you are in the country, snake fungal disease in that area can be more closely related to snake fungal disease there than in another area of the country.
Mm-hmm.
And also there's snake fungal disease in Europe, but there hasn't really been snake fungal disease documented in South America to this point. It's been documented in China recently. I think they found it on a constrictor species there. I think it's been found in Japan recently, but it's not Taiwan. Yeah. But it's not found. It's not ubiquitous geographically. Like there are still regions of the world where it hasn't been documented.
Which is possibly just due to lack of detection, like lack of looking for it. Yeah, and not like true absence.
Yeah,
possibly.
Yeah,
I think it's interesting that you guys had mentioned that those photos that we just posted, a lot of them almost seem like some kind of scarring or battle wounds from predation or from fighting prey in search of food, whatever. And I've seen a lot of animals that in the wild and in captivity that had scars like that, and it's almost like you wouldn't even think that it's a fungus. Does that make sense? And I know that the only time that in my earlier years doing this stuff, the only time that I ever remember people talking about a quote unquote snake fungus, this is way before there was, snake fungal disease, was people that get like water blisters from bad husbandry, or, snakes that need to be a little more arid and they've got these sores in their sides, and it's clearly from bad husbandry. But I remember vividly that maybe 15, 20 years ago, there was a lot of bushmasters being brought in from Suriname, French Guyana, and the rule of thumb was you had to keep them at a high humidity, but with no moisture on the ground. And people would say, well, how do you do that? And, there was different ways of having different gradients with, multiple humid hides and multiple dry hides. And basically what happened is if the substrate was too moist, they would get a fungus on their pectoral scales that was like in between each scale. And they used to call it the bushmaster rot. And basically if your bushmaster got that pink inflamed underbelly, it was toast. It was done. And people would soak it in chlorhexidine. They would put, prescription athlete's foot prescription cream on it, and nothing worked. And the snake wound up passing away. But then there was kind of a lull because people figured out, okay, if I do X, Y, and Z, I won't get that disease. And then out of nowhere, we have snake fungal disease. So I didn't know there was some kind of correlation with that in some regard.
Yeah. Well, that's interesting. That's actually the first I've heard of that. And because of that, I can't really comment as to what could have been causing that particular disease, or if it might be directly related to snake fungal disease. But I can say that one of the things that I talk about often when I give talks is there's a paper that came out in 2012 by a guy named Fisher, who basically showed that over time, the amount of diseases that are caused by fungal pathogens in wildlife has been increasing over the 21st century. And that's something that's worthy of concern, particularly to wildlife biologists, because when you look at the proportion of different pathogens that cause actual extinction or extirpation in their hosts, like fungus versus viruses or bacteria or helminths like worms, things like that, fungi disproportionately, by 40% or something like that,
wow,
cause way more extinction and extirpation in their hosts than other pathogenic organisms do.
Wow.
So, yeah.
Is that just from the lack of building up sort of an immunity to it like they would with viruses and bacteria? Is it?
Yeah, it's a great question, and one that I don't know I certainly don't have a great answer for it, and I'm not sure that as a scientific community we necessarily have a great answer for it. Donnie might have some thoughts on it. I think you're muted.
Yeah, you are.
Not only do I log on late due to but mute myself midway through, I also forgot my beer. And I'm sitting here watching you guys drink and smoke, and it's really making me jealous. But
well,
yeah,
I asked Alex. I said, "You don't got like a fifth of bourbon in the desk drawer?" And he says, "No."
I mean, I do right here.
There you go.
But
You're saying, Doc.
yeah. But I was going to speak a little bit more about like the Bushmaster rot side of things. Like this fungi, like all fungi, typically like wetter environments, more humid environments, and this particular fungus has a suite of enzymatic activity, so it's able to produce keratinases and lipases, right? So the skin of snakes is made primarily of keratin, and then also a variety of different fatty acids and lipids. And so this particular fungus specializes on snake skin and probably likes moist conditions. So I could see when you alter the environment for the Bushmaster and end up getting this fungus growing on its scales. It doesn't surprise me all that much. I can imagine, I don't know much about snake husbandry, but I can imagine it's probably pretty challenging to manage that type of environment in general and suppress fungal growth.
Some more than others. Yeah.
And let's, and not to, say that chlorhexidine is the end all be all, but it really does wonders for as weak as it is.
Mm-hmm.
And I mean, even with some of the I don't want to say normal mold, but like some of the white fluffy stuff that you get with some of the bioactive enclosures, or old rotten oak leaves. We love using oak leaves in enclosures, and before you know it, if there's not good ventilation, you got white fuzz. And some people have macroscopic organisms to take care of it. Other people, you just squirt a little chlorhex on it, and boom, you're in business. Well, until you have more spores.
Growing in my dart frog cages sometimes.
Yeah, there you go.
Would pop up, and then they'd hang out for a little bit, and then they'd disappear, and then a couple more would pop up, and then they'd disappear.
Yeah, but it is challenging with something like those Bushmasters, because you have a baby pit viper that is extremely stress prone, and you're trying to leave it alone and give it the best husbandry you can, but it took a lot of guys and gals, unfortunately, a lot of losses to learn that different humidity gradient with those specific animals. So
that is interesting. Yeah, not surprising. I can imagine you probably. Go ahead.
Say it's a fine.
Yeah, I was going to say, like, I can imagine, like, if you're getting I guess I'm surprised at the fungus being present in an enclosure in the first place, unless it came with the animal. We think that this fungus is capable of surviving in substrate, and there are some documented instances from our lab showing that it does survive in the soil. There's another recent documented instance showing that it survives in hibernaculum soil, and then a variety of like Petri plate tests showing that the fungus can grow in soil substrates. But like, I don't know. I just think, like, if you're building an enclosure, either the fungus is coming with the snake, which is most likely, I'd say, or alternatively, you're scooping soil or building like a natural habitat from oak leaves and stuff like that, and you're bringing the fungus which seems to me to be less likely.
Well, most of us back in those days, I mean, even still today, let's be real, let's be frank, we use bags of cypress mulch that we buy from Home Depot. And some people are a little more neurotic than others, and they'll bake it on a cookie sheet in the oven at 200 for however long they do it. And then other people, honestly, like myself, for the longest time, I just dumped it in the enclosure, and not realizing that you have this extremely humid, festering bag of slowly decaying wood, and sometimes there'd be bugs in there. Sometimes there would be straight up mold in there, and you throw a couple of those pieces out and you scoop out that top layer, spray some chlorhexidine and keep on trucking. And I feel like it goes back to us not knowing the cross contamination that we were doing back in the day. We didn't know. So, and then at the same time, you have these animals like these bushmasters that were coming from an animal exporter in Suriname, where they're keeping, birds, turtles, amphibians, snakes, mammals, all in the same warehouse, and who knows, this could be, something that comes from bat guano for all we know, who knows?
Mm-hmm.
Well, the thermal maximum of this fungus is about 35 degrees Celsius. So if you're baking, you're probably not a bad idea.
That's good.
And
That's good
have
to know.
you, knowing that, have you exposed animals that are infected with it for an extended period of time to see if that knocks a lot of it out?
That was kind of what we were attempting to do with thermal gradient, like giving them a range of temperatures and letting them see where they chose to bask. I remember correctly, I'm pretty sure I did. They weren't choosing like the hottest temperature every time or anything like that.
Mm-hmm.
I'm almost positive that was the case because I'm pretty sure I'd remember if they were all glued to one side of the enclosure throughout the experiment.
That is the case. Mm-hmm.
Yeah.
Huh.
Sidecar to this is the fungus that we're speaking about. Forgive me for not remembering the actual species, but is it, I don't want to say endemic to snakes, or does it grow on other things? Can it live on other things? I mean, yes, let's say the spores are dormant in pieces of damp, rotting cypress mulch, right? And now all of a sudden it's been waiting for the perfect opportunity to hop on that garter snake. Or is it still growing? It just isn't growing as well, or it's not producing spores as well as if it was on a living host.
Yeah, that's a great series of questions. So it definitely can grow in soil. So our lab has found it on soil when we went looking basically for its DNA. And then other labs have also found, for example, that it's more prevalent inside of snake hibernacula in the soil than like in the soil right outside of snake hibernacula. So it grows in soil, but my guess would be that it grows in soil more as it's a reservoir. It's looking for a host and kind of just maintaining there rather than that being a primary means by which it maintains itself on the landscape. It seems to, on the landscape, basically only affect snakes. I don't know of any records of it infecting something other than a snake on the landscape.
Actually, so at MSA, the Mycological Society of America conference, we just saw a poster showing that it can grow on lizard. I can't remember which species of lizard, but it was an inoculation experiment and not a natural.
Right. That's what I was going to say.
Mm-hmm, yeah. Sorry.
No, it's okay. Yeah, that's what I was going to say. It doesn't, as far as I know, as far as I've seen, it's never been reported to infect anything but a snake on the landscape. But at the Mycological Society of America, someone demonstrated that they were able to experimentally infect bearded dragons with it. But, bearded dragons haven't been exported from Australia since, you guys would know better than I would, but it's been probably 20 or 30 years now.
Yeah, the late 80s.
Yeah. So, I mean, it's probably not the best necessarily model organism because of the fact that they're relatively inbred in captivity at this point. And just because you can induce something in a laboratory conditions doesn't necessarily mean that it's taking place in a landscape. So it can be done, but whether it's actually occurring in nature is another question.
Yeah.
Yeah, we've done some microscopic work with the fungus, like looked at what it's actually doing in soil and haven't documented sporulation. We can see, so like the vegetative state of a fungus is called like the hyphae or the mycelium. And so it's growing. It's like that white fluffy stuff that you see sometimes in your enclosures. Mm-hmm. It's growing and extending throughout, the soil, but we haven't seen documented sporulation of the fungus. So yeah, it's tough to say whether it's hanging out and, in dormancy, waiting to colonize something that's enriched in keratin and lipids, or if it's just kind of living and surviving like at a low level on reptile skin. I think we just don't quite know yet.
Yeah. Is it affecting immunocompromised snakes more so than sort of normal, healthier snakes that aren't struggling? Like, do you notice a much more serious case with snakes that are already having issues, immune system wise?
Do you know of a study like that?
I don't. Yeah.
I guess it's hard to ask because, like, we know that there's a response to viruses and bacteria and stuff, but as far as like fungi goes, like I don't really know how the body reacts to that. I know, like, we get toe fungus, we throw some Lotrimin on it, no big deal, whatever, it's gone. But I don't know if there's a similar reaction from the immune system to something like that.
Yeah, to be honest, I think there are similar reactions in snakes to fungi compared to like viruses or bacteria, but I don't know of a study that has investigated that specifically.
Yeah, I imagine it'd be difficult because you'd essentially need an extremely healthy animal, and then you also, or animals plural, and then you also need some like anemic stuff, and I imagine that would be quite difficult to reproduce, if that makes any sense.
I can think of like at a population level some things that might be relevant. So one concept that's talked about in conservation biology is the extinction vortex, and so this kind of ties back to one of the original kind of reports of snake fungal disease of timber rattlesnakes before it was even really known that it was snake fungal disease. In fact, in this paper, they weren't even able to determine what the fungus was, but now it's kind of commonly accepted that it may have been snake fungal disease just based on the timing and the symptomology and things like that. And this was in New Hampshire, I want to say. I'd have to double check the exact state, but it's definitely that's correct. Yeah. And so this was the last remaining population there. It was a year that had very high rainfall, and also this population was already known to have low genetic diversity because of the fact that it had been reduced to a low level. And so all of these factors together is what was assumed or was hypothesized to cause this, like, one particular year where this fungus caused a decline around 50% in this population. And so that's where you get this like vortex effect of like the population is already declining. There are several factors that make it vulnerable. And then a disease kind of comes in because the population is already unstable or suffering as a result of these other effects. And one thing to kind of keep in mind is that if you have a contiguous landscape, so, one of the things you're talking about with bush masters is this disease is that, like, if they don't have a gradient, then they have a harder time responding to bushmaster rot. And if you cut up a landscape and you fragment it, you degrade different habitat types that are available within that matrix, wild animals are losing availability to kind of remediate or respond to disease in the same way that if you provide like a captive animal less options in their enclosure, they also aren't able to remediate diseases effectively. So that's another aspect where it's like, as you degrade a landscape, your populations aren't able to respond as effectively.
Mm-hmm.
Very interesting. Very interesting. You had mentioned that they were taking, or you guys were taking, I don't want to say, for lack of a better word, soil samples from the hibernaculum. Were there, I don't know if you'd want to even call it a hibernaculum if there's no animal, hibernating in there, but when you guys did this, was there ever samples taken from what may have looked to be like, when you're herping and you find like, oh, there's going to be something right underneath this log and there's nothing there. Did you still take soil samples? Was there, is it only found in, dens or hibernaculums or rotten logs, whatever you want to call it, that had inhabitants? Or did you just check everything? Sorry if that was a little space kiddy there for a second.
You're fine.
No.
We're very simple people.
No.
We have simple questions.
Yeah, so we actually did not conduct the hibernacula soil reservoir study. That was another lab, but our lab did conduct a study where it was a widespread landscape study, and Donnie can speak about this more than I can because I was not. The study was conducted before I joined the lab, but at the time, one of the questions that was being asked was, does the snake microbiome differ from the water and soil microbiome? Because at the time that was actually an unknown quantity, because it could have just been that snakes basically have the same microbiome as the soil. They're hanging out on it all the time.
Yeah. So we collected soils and water from point capture locations, kind of with the thought that if the snake is living in that particular home range and infected with ophidiomyces, that maybe it's shedding some of that fungus into its environment, and the fungus is colonizing that particular environment. And so you could see a signature of the fungus present in the soil or the water or leaf litter, whatever we were sampling. And we did find several point capture locations, the soils that we basically just kind of swabbed in circles, and picked up some of the soil, put it into a tube, and extracted the DNA and used a quantitative PCR test to determine presence or absence, much like the COVID tests that are conducted. Yeah, so we did find it in the soils, but we did not find it in the water, which is kind of odd because in the same study we actually, and since we've documented that water snakes seem to have a higher prevalence of the fungus, but we weren't finding it in like liters of water that were collected, filtered through this really micropore filter, and then DNA extracted from that. So it's kind of interesting and suggests that it prefers to live as a saprophyte or something that's decaying, like dead and decaying material, when it's not living on the skin of snakes. Although I do think that the enzymatic suite and the host specificity, being that it lives and grows and is found most of the time on snakes, suggests that it's probably pretty typical to snake skin. I think probably more so than the soil environment.
Wow. That very interesting, especially since like Nerodia and ribbons and garters and that sort of thing, they're so aquatic. It's interesting. It's very interesting.
Yeah, we see a lot of lesions. I think, Alex, you would probably agree with this. Like, just like being in the field with you, I feel like, I don't know, maybe every fifth snake we came across, you'd see a lesion, something that was suggestive of a clinical sign of snake fungal disease.
Yeah, I feel like that's definitely a fair estimation. It might be higher than that, but
Nerodia, same thing with ribbons and garters, like ribbons in particular, like out West Tennessee, where you catch them a little bit more often. I'd say, maybe even 1 in 3 or 1 in 4 you'd see.
Yeah, that's
something
more for Nerodia than terrestrial snakes, but yeah.
Man, it's gonna make me really check every snake I find now. Because I mean, it's so many times I just chalk it up to, it got a scar because it tried to eat a fish and the fish bit it back, or
cats, whatever.
Or baby alligators, turtles, whatever. But,
if you're interested in citizen science, or any of the listeners interested in citizen science, they can totally reach out. We routinely send out snake fungal disease sampling kits to all different places across the country. That if people are out herping, collecting snakes, and they want to do some swabbing and send us in swabs, we're happy to process them.
Yeah, absolutely. I would 110% do that.
Is that animals that have lesions, don't have lesions, both?
All of the above. Yep. Any snake swab is a useful swab.
Oh man. I mean, there's some nights I go out and don't find anything, and there's other nights, we'll find 30, 40 water snakes in a night, so very interesting.
That's like Carolina herping, right?
What's that?
I said that's Carolina herping for you, right?
That's South Florida.
Oh, South Florida.
I'm way south.
So I don't find anything, so I don't even bother.
Yeah.
I did see a dead copperhead on the road the other day, though. So
nice.
What about like dead snakes? You stop.
We salvage dead snakes as well. It's typically for other types of studies and for like educational purposes. Like we routinely host student groups on campus from local community colleges. They come and they'll visit, and we'll lay out a bunch of dead snakes that we have frozen, and we'll teach them like snake morphology, how to swab snakes. We'll set up a bunch of Petri plates and see what microorganisms grow on them, and a variety of different things just to teach about snake biology as well as the microbiome and disease ecology as well. So yeah, we do take dead snakes as well, as long as they don't stink. That's like you got to sniff them before you send them.
Yeah, well, I imagine if it's that far gone that start to get funky, it probably isn't the most conducive to the project.
Falling apart as you're putting it in a bag.
Yeah, you don't want that. Yeah, when they're flattened, when you're peeling them off the pavement, it's probably a bit too late.
Yeah. Now, have you guys seen with any of the control animals or the stuff that you inoculated where it developed the for lack of a better word, the blisters or whatever, and then through husbandry has righted itself and has been cured, so to speak?
Yeah. So we haven't had any control animals that we inoculated that developed. Well, I don't think we ever had a control animal inoculate blister or develop blisters as a result of our inoculation protocol.
Okay.
But this actually, I was kind of hoping to segue a little bit into this when we started talking about swabbing. So our lab, we mentioned the fact that we do kind of microbiology of herps and stuff like that. So a lot of with those swabs, we look at snake fungal disease, but we also look at the microbiome or the bacteria that live on the skin of these animals. And so, for example, with the work that I did over the course of my master's thesis, we brought snakes into captivity, and those were animals that we were looking at their microbiome for. And we did that because we thought it would be most informative to have animals that had a wild microbiome instead of these were corn snakes that were raised in captivity. So how informative is it to know this is how snake fungal disease is affecting the microbiome of those animals? Yeah. And so even though we initially screened those animals for snake fungal disease, a few of our control animals developed snake fungal disease, and so we didn't include them in any of our analyses or things like that.
Yeah, but the clearing of disease, I don't think we've seen. Alex, you could probably speak of this. You were watching these animals day in and day out and documenting disease development, but I once you typically give them the fungus, they develop signs that are recognizable as snake fungal disease and don't seem to clear it up. It's been shown, even on the landscape, oftentimes in our work and others' work, you swab for snake fungal disease and the animal looks perfectly healthy, but it will have a signature of the fungus being present. And so I don't know if that's just long-term infection and it's just persisting at a level that's undetectable by the eye, or perhaps maybe it's subcutaneous, beneath the layer that you might be able to detect with the eye, and you're actually getting at it using that swab or something that we just don't yet understand, which is equally likely.
And do the blisters have any hand in spreading more of the spores? Do the blisters have some sort of physical advantage to carrying it on more? Is that a reason that they're there?
Yeah. The fungus does sporulate on dying, dead necrotic tissue. Yeah, okay. It does. Mm-hmm. Yeah, oftentimes, if you look at some of the literature, some of the pictures that you were showing earlier, you can see the fungus producing these conidia or asexual spores in some of the histopathology images, it's thin slices of tissue, and researchers typically stain the tissue, and you can actually see some of the spores being produced and the hyphae growing through the tissue.
Wow.
Yeah, it's pretty neat. Not stuff that we do in the lab, but it makes for some really nice images.
Yeah, it's incredible.
And understanding oh, go ahead.
No, I don't mean to cut you off. Go on. I'm sorry.
Oh, I was just going to say, just kind of understanding the biology of infection, the pathology of the organism as well, how it grows and invades subcutaneous tissue into deeper parts, more visceral organs and whatnot. You can see with those really nice histopathology images.
Nice. So my question is, have you noticed in your captive animals that have it an increase in shedding at all?
Yeah, that's what I was going to ask too, because I'm curious about that.
Well, this is all Alex. This was all his idea. He had this really, really cool idea. He came to me one day. He's like, you know what? I went home after being in the lab today. I drank a ton of coffee and I sat in front of a whiteboard for I don't even know how many hours. And he had this awesome idea and laid out an entire study for me. And I just said, that's a fantastic idea. You should do that for your masters. So he can tell you about that. That's like part 2 of his masters that he's still working to publish right now.
Hell yeah.
Yeah, it's one of those things that is on the back burner just because we didn't get a ton of samples from it, but it will get out at some point. But one of the things that I became interested in, so Donnie was talking about this histopathological images, and there's one in particular that's really cool where you can see there's like a layer of snake skin that's about to be shed, and then there's the new layer that's about to come in, and you can actually see the fungus proliferating from the top layer into the new layer. So even though it's about to be shed, there's going to be remnants of it in that new skin that's going to be retained. And that got me thinking about like, how does shedding affect this process? Like, and also how does shedding affect the microbiome? Because when the snake sheds all that skin, like where do the microbes go? And so I wanted to basically understand, as the snake sheds its skin, are there any microbes left, or does it shed its microbiome every time it sheds its skin? And that was a bad idea to suggest to Donnie, because it meant that to actually sample that, I had to be there when a snake sheds its skin. And you guys know from keeping snakes that you see that maybe, even if you have a lot of snakes, you see that really only every once in a while. And so the way I decided I was going to study that was just to have me and then another undergrad there basically all the time, always watching them, waiting for snakes to shed. But one thing that was cool that we found out by doing that is, so I'd be in there with a red light going through every like 15 or 30 minutes looking at them.
You should describe your lifestyle change.
Well, I went from being a normal working human to someone who lived only between the hours of, I don't know, like 8 o'clock at night and 6 o'clock in the morning. Yeah, that was my life. Just being up in the middle of the night, walking through a dark room full of 24 snakes with a red light, waiting for them to shed. But when snakes finally started to shed during the study, they would only shed during the day, like never when I was there. And I was like, this is absurd. And
just a big middle finger to your whole plan.
Right. And by the third or fourth, but there was an undergrad there to sample it, so we were getting the samples, but it was just that it was like when I was never there. And eventually I thought about it, and I was like, I think the reason they're shedding during the day is because they're freaked out by me coming around with the red light at night. And when they're during the day, they feel more chilled because there's just someone sitting in their room. So by the end of the experiment, we took the cages of snakes that we knew were in pre-shed, because they had cloudy eyes and stuff like that. And we put red lights on either end of it and set them to automatically flick on and flick off every 30 minutes. And sure shit, those snakes also shed during the day. And to realize that you're like, how many hours of your life could have been replaced with like $30 of the hardware from Home Depot, pretty hard hitting.
That's rough.
Yeah.
Science.
Yeah, right?
Yeah.
No one told you they were diurnal.
Yeah. Well, I honestly thought they might have shed at night, just because water snakes and stuff like that. Like even if the lights weren't there. But I published a note on that, at least in like just a little local herpetology journal. So we have that data set basically of it's like 5 or 6 samples of snakes where, like, as the skin, like in some of them we actually peeled the skin back and was able to sample immediately. So we can see, like, right at the point of a snake shedding, what does the microbiome look like, and also how does that affect snake fungal disease? Mm-hmm. And I've done the analysis on the snake fungal disease portion of things and found that we didn't see a significant reduction in the amount of pathogens just from a single shedding event. And so other authors have suggested that, and shown that snakes have these rapid periods of shedding after they get snake fungal disease, and that's probably causing if snakes clear infection, it's probably because they shed rapidly And that would seem to be the case based on the work that I did, where it shows that like the snake shedding once doesn't necessarily reduce the amount of fungus that they have, probably because it's already grown through that layer. But if they shed maybe 2 or 3 times in a rapid period, that might actually do the trick.
Mm-hmm. Yeah, I mean, I asked about the shedding just because I know, or I've seen, I mean, it's purely anecdotal, but I've noticed if there's snakes that get stuck between tubs and shelves or something, or they get some sort of like damage, they almost immediately, within the same, a handful of days of that happening, they go into a shed cycle. Like their body is automatically like, I got to fix this.
Yeah, like from a mouse bite.
Something. Yeah. And so I wondered if that would be almost like a continuous response to that, is to constantly go into like shedding and some sort of, again, an immuno response to getting rid of it. But I mean, are they even having good sheds at all? If they have it, if it's really bad, is it just an absolute nightmare, or is it full complete sheds still?
I think it depends on how bad it is. If it gets really bad, there's definitely portions of the skin that don't shed well. And then, they're like the portions of the skin that aren't shedding well, you're retaining the fungal tissue from it. And so then it's not really effective. It's not a good mechanism at that point to clear infection. Mm-hmm. But early in infection, it seems to shed pretty well. And you can see like when it sheds, there will be regions that look like they have the dermatitis or they have necrotic tissue on them that are shed. And so I think sometimes it depends on like how far is the fungus proliferated already, how bad is infection, things like that.
Well, we have a question in the comments. Our good friend Henry the Hen Dog, he specializes in Ophiophagus snakes. And his question is, what happens to a snake that eats a snake with snake fungal disease?
Yeah, that's a good question. I don't know if there's research on it, but I can give you some anecdotes from the field.
Yeah, of course.
Which is that Lamprobeltis in general do seem to carry a pretty heavy burden for snake fungal disease.
Okay.
And that there are a couple of sites that I go to early in the season where the black king snakes always seem to have a lot of snake fungal disease.
Interesting.
And those are sites
it makes sense because they're grabbing snakes and they're wrapping them and they're in contact with a lot of surface area of that body that would have it. So that makes total sense.
Yeah. I mean, that's not based on any scientific rigor at all.
An observation.
Yeah. It's just an observation.
Yeah. Excellent. That's a
cool thought.
Yeah. The thing that I found the most disturbing about it all when I was reading about it, was just the fact that it was like, yeah, we found it up in the Northeast, and then like a couple weeks would pass and be like, oh, they found it out west, and then it'd be, oh, they found it down in Georgia, oh, they found it, in California, and it's like, I'm assuming, I mean, there's no way it would travel that fast, but is it safe to assume that it was just already there and now people were looking for it, and so now they're finding it?
Yeah, that's what I always thought. I always thought it's been there, and obviously I'm not a scientist, I'm not a field biologist or a field researcher or anything, but it just made sense to me that was maybe human encroachment and agricultural runoff and things of these sorts, global warming, whatever you want to call it, is obviously a contributing hand. But it was, to my personal opinion, that now everyone knows what they're looking for.
Right.
And the same way that I thought it was a scar, or I thought it was, an injury from, wrapping a prey item or something. And, it's just a scar, it'll heal, whatever. And because it's in captivity, it's living a good life. Maybe it didn't have a chance to really do more damage or something. Who knows? Mm-hmm. But now we know what to look for. So I don't know.
Well, yeah. Very similar to the sort of the Nido thing, where it's like, oh, an animal has a respiratory infection, okay, it happens. They get them. Like, we noticed that in green trees, like, yeah, this time of year it gets a respiratory infection. We don't know why. And then once they find out that it's a virus, now it's like, oh yeah, I tested and I have it. Oh yeah, I tested, I have it. Like, yeah, I got it too. And like, now that they sort of know what I guess it is they're looking for, and it sounds like it's very similar to how it was up in the Northeast when they were originally seeing these animals coming out of hibernation like this, that maybe now that they see and have, like, they've identified sort of the culprit, like now everyone else is sort of seeing it too, and they can sort of pinpoint it and identify it as well.
Yeah, I think one of the things you guys raise a great point, which is that like all of a sudden people around the country started looking, and so you can't find if you don't look, Mm-hmm. And another, so we were talking about hibernation blisters a little bit before. And since then, there's actually been retroactive work to do DNA extraction and then keep PCR on some of those hibernation blisters. And it's been found retroactively in museum specimens so animals that were collected a long time ago have Ophidiomyces. So that's really good evidence that it wasn't something that was introduced in 2001 and we didn't notice it until 2010.
Right. Wow. That's awesome.
Yeah, it's
kind of reassuring,
yeah.
in a grotesque way.
Yeah. And then there was actually a paper that came out this year by a number of people. And that paper showed that based on the divergence time of the different clades of Ophidiomyces, like we were talking about before in the United States, it suggests that Ophidiomyces may have been introduced to the United States between 100 and 250 years ago. So it was likely brought from Europeans into America, which is kind of interesting because it still persists in Europe as well. I was actually just talking to someone who studies Ophidiomyces and grass snakes in the UK at a conference, and he was telling me that all of their grass snakes, I think 98% of the lesions they recorded, 98% of the lesions they recorded were on the ventrum, which is really unusual. I mean, in the US, we get them all over the body on the nasolabial pits and vipers, things like that. Whereas in grass snakes, he was saying he basically only gets them on the ventrum of snakes. So there are some differences between what it's doing to European snakes and what it's doing to American snakes. But it seems based on recent analyses that Ophidiomyces was something that was introduced to America a while ago and is now basically being released as a more severe disease-causing organism, probably due to the fact that there's more habitat fragmentation, there's more agrochemical runoff, things like that.
Things are getting more and more isolated.
Right, that extinction vortex that we were talking about before.
Yeah. You build a highway and you change everything.
Right.
Yeah. It's interesting that the nature scenes are only ventral, and we see the Neurodia and Thanopsis or whatever. It's all over them. There are pit vipers on their head and stuff, but it makes you wonder if it's environmental because of whatever foliage or whatever, microbiology is in the European soil opposed to our soil, vice versa. Has anyone ever looked at from those museum specimen jars or what have you? Is there any kind of, I don't want to say evolution, but do you feel like maybe it is, for lack of a better synonym, evolving from something different? Could this have been a mammalian fungus that has adapted to, well, the mammals are washing themselves, so we'll latch onto a snake? Does that sound crazy?
Well, their most recent ancestors were definitely also reptile pathogens because they're nestled within this clade of fungus that also infect lizards and things like that. It's kind of a clade of specialists of reptile pathogens. But in the larger taxonomic context, they're a member of this group, the Onigenales, which are also pathogenic fungi, but they have a much broader suite of hosts taxonomically, so they'll infect some human pathogens are actually related to that clade, things like that. So often, when people know nothing about snake fungal disease and I'm trying to introduce them to the topic, I'll be imagine a really bad athlete's foot and imagine if you got it on your face. That's because it's not that far of a jump taxonomically to describe it that way.
Very interesting.
I don't understand what the sort of the ecological purposes of something like that, because I think about that too with things like, I don't know, something considerably larger like a gnat. We have sand gnats here and they're brutal. And I'm aside from feeding tiny spiders and other insects and stuff, why do you exist? Why are you here? And with stuff like this, I have to wonder the same thing. It's okay, you attach yourself to a snake and you get spread somewhere else, and that's kind of it. Now it's wash, rinse, repeat. So maybe Donnie can answer this. What's the purpose of something like that? What function does it? I
lost you just a little. I lost you a little bit, but I think I got the general gist of the question. Yeah, so this fungus is primarily known as it's an asexual fungus, not producing sexually. And so, like Alex was saying, it definitely specializes in a particular niche, and that niche is a keratin-rich a keratin-rich substrate. It grows extremely well on keratin and specializes on keratin, and it doesn't need to reproduce sexually. There's very little genetic diversity. It's happy living and surviving in that particular niche and producing millions and millions of asexual spores to spread into the environment and into neighboring snake skins because it specializes in that environment. That is its ecological niche. It's happy there. It doesn't need to go anywhere else.
Yeah, I imagine it's just another one of Mother Nature's, control devices for whether it be population or checks and balances or what have you. And, you throw humans in where we take up so much more, and we were talking about isolation, that who knows?
Unnatural selection.
It is worth keeping in mind that even though it's probably been here for a while, like our native snakes are not really designed to interact with this disease. Even if it got here 250 years ago in evolutionary timescales, that's still a very brief period of time.
Right.
Yeah.
Very interesting.
Patrick brings up a good point. He said, can humans get it on their nails or their hair? Or is that a different sort of type of keratin that we're talking about?
It hasn't been documented.
Not that we know of.
Yeah,
Phil, we should try it with your beard,
a fair amount.
Let's get someone rubbed on Phil's beard.
Well, I mean, it's interesting. We joking about my beard, God forbid. Let's say, because we're talking about these gross hypotheticals, I am field herping and I find a pygmy rattlesnake and I throw it in the bucket so that I can tube it and swab it with one of your samples, right? And I do everything kosher, and we're done. And I mail it off and I let the little bugger go, right? I need to clean that bucket. I need to clean those tubes. I need to wash my hands, especially if I'm catching more animals or, God forbid, bringing it home to my collection. That cross-contamination, I imagine, can in the blink of an eye, right?
Mm-hmm. Yeah,
absolutely.
Is there a particular method for, especially in a more, I don't want to say sterile, because your snake room at your house is not exactly the most sterile, but is there a choice way of reducing risk of contamination at home?
Yeah. There was a paper that came out in 2016 that looked at the effectiveness of different disinfectants on basically preventing OO from growing in a petri plate after it had been sprayed. So they would create a solution that they could use to inoculate plates, use this disinfectant on it, and then see how many new colonies would form after it had been sprayed with this disinfectant. And so they tested some household cleaners. I think Simple Green was one of them. They tested chlorhexidine. They tested two different bleach concentrations of bleach, and they also tested 70% ethanol and either 5% or 3% bleach or 70% ethanol were your best bets with a 2-minute contact time. The caveat being with both of those disinfectants that you should make them fresh basically every time you use them, because bleach degrades over time, and then 70% ethanol can degrade as it evaporates. So if you really want to be careful, those are what you should be using, but you should be making them fresh whenever you need to disinfect. So if you're going out into the field, if you're going to be wiping down your racks, if you have a new animal that you're quarantining, things like that.
Yeah, that's brilliant. It's good.
Yeah, and I mean that's something a lot of people don't really consider, is the hooks that you carry around your car and you go herping with all the time, disinfecting those, just so you're not introducing other stuff, if not SFD or something into other populations or pockets.
Yeah. If I could make a plea about that, I love field herping. I would encourage people to do it as amateurs. If people are interested in doing it as amateurs, I'd certainly say it's not something that only scientists should be allowed to do, because it gets people interested in the field, of course. But that's super important, because not only is it about things that we know are out there, like chytrid and snake fungal disease, but RO, the pentastone worms in Florida, we didn't even really know they were a thing until a few years ago. And there's probably things that are out there right now that we're not even really aware of that are spreading. Mm-hmm. So it's super important that when people go out into the fields, they disinfect properly, and that you spray the mud off your boots before you disinfect them, that you scrub, so that way what you're disinfecting actually reaches the hard surfaces of whatever you're disinfecting, not just I sprayed some mud, but then there was inch of mud caked on there. So the bottom of it, like, who knows? And then I went and traipsed around the vernal pool, you know?
Yeah. Now, is that something like we were just talking about keeping snake hooks in the car? Like I keep snake hooks in the car. If I'm road cruising and I come across, 4 rattlesnakes in a night, I'm going to use the same hook. Should I be disinfecting the hook in between each find? Or is it something like, well, I'm on the same road within a 3-mile span?
Ideally, you should disinfect it between each find, just because, depending on whether you're visiting hibernacula and stuff like that, you might know that like different snakes from the same area could be using different hibernacula. They might not necessarily be interacting with each other. Even if they're using the same hibernacula, they might leave in spring and not see each other again for 4 or 5 months. So I just feel like it's a best management practice to like disinfect between every animal that you interact with.
Mm-hmm.
Yeah.
Okay.
Wipe your hands down, have some Lysol wipes.
Yep.
Wipe your hands down in between animals. Yep. Spray down your tubes, your bucket, whatever it might be.
Yeah, anything the animal touches, if you just in the tubes, disinfect the tube. Like Donnie said, if it goes in a bucket, disinfect the bucket and do it before you leave. So that way, next time you cruise an animal, you're ready to hop out and do your thing. Because I know there's no patience when I see an animal on the road.
right. Yeah, well, you don't have time.
Exactly.
Yeah,
I took someone out for the first time with me on Friday, and they were like, "I can't believe how fast you got out of the car." And I was like, "Welcome to the show." Like,
takes time to learn, develop this. Break and throw it into gear.
Or don't throw it into gear.
Let it keep rolling. Tell them to grab the wheel. I'm gonna jump out. Does UV do anything to fungal infections like that? Does that have any effect on it, like it would bacteria or viruses?
Yeah. You can kill fungi with UV light, but I can't imagine you could treat an animal with UV light. I don't think that's possible. And I don't think it would be a very effective way to disinfect equipment, for obvious reasons. But yeah, UV light in general is effective at killing both bacteria and fungi given the appropriate length of exposure time.
Mm-hmm. Yeah, I mean the whole thing's just bizarre, especially considering how it seems like it popped up everywhere all at once.
Yep. Yeah, I think it's once people start looking, I think it's going to pop up more and more. I was just on the phone today with Ryan Hanscom out at San Diego State, and he's telling me he's out in Nebraska looking in rattlesnake hibernacula, and as the animals were emerging, and many of them were coming out with clinical signs typical of snake fungal disease.
Really?
And unfortunately, I don't think it's been documented in Nebraska yet either, not that I recall. But
the Southwest is kind of a black hole for snake fungal disease. I know Nebraska is more northern, but I just feel like in the Midwest in general, it hasn't been reported a lot. And that what would that be? Longitude, I guess.
Yeah.
I guess it's not necessarily temperature sensitive if they're finding it, up in the Northeast.
Yeah.
Have they done any studies there as far as like cold being more effective than heat or anything like that? Like, can it survive freezing temperatures? I mean, if it's in a hibernaculum, that's going to be slightly warmer than whatever it is outside. But does it go dormant?
We freeze the fungus to negative 80 degrees Celsius, and then we can grow it again. But it's using a pretty specialized technique when we do that. But yeah, it has a pretty broad thermal range. I think it grows up to approximately 35 degrees Celsius. And down, I can imagine, to pretty close to freezing. It'll probably still grow, but I'd anticipate it being able to persist a freezing event.
So going back to like our buddy Henry that has multiple chest freezers full of dead snakes to feed other snakes. If he has something, say, below zero for 90 days, do you think that would be considerably safe, or there should be other precautions to be taken?
Oh, I don't know if I feel qualified to answer that
fair
question.
enough. You said enough right there, sir.
Yeah, I mean,
duly noted.
Yeah. I do think that you'd be able to detect the fungus. You'd definitely be able to detect the fungus if it were present on those dead snakes. Whether it be viable or not is another question. It's kind of a cool idea, cool little study that an undergraduate or maybe even master's student could do.
Yeah.
I guess that's another good question is, does it, if it is on a snake and that snake dies, do you see a larger sort of batch of it on a dead snake because now it doesn't have that immune system, at least fighting back a little bit? Do you see it more prevalent in dead snakes than the live ones?
I don't know if we find many dead. I don't think I've ever found a dead snake and then sampled it like with that thought, like it died recently from snake fungal disease and now the fungus is colonizing it as like a decay fungus instead of as like a pathogen. But that's an interesting thought.
Yeah.
I don't know. What do you think, Alex?
I don't know. I think it's an interesting thought for sure. I'd be curious to see if there's like a tipping point, like recently after death, if it's like now the fungus is like, bouncer left the party, like let's go nuts. But but for my thought would be that it can't compete. After a certain point, my thought would be that there are organisms that are better at decaying a dead snake than because you got to figure it's got to be somewhat specialized to be able to do it while the snake's alive.
Yeah, I would agree. Yeah, at that point, it's probably the opportunist party more so than Ophidiomyces.
Do they notice any difference with, like, timber rattlers, for example? Do they seem to get it more in certain areas in comparison to, like, colubrids? Because it seems like a lot of the pictures I've seen of colubrids, like, they seem to get it really bad in the face and the eyes. And then with timbers, from what I've seen, which isn't a ton, it seems like a much sort of, not necessarily milder, but it just doesn't seem to look as extreme as I've seen on some colubrids. Is there any correlation there? Is it?
Yeah. I mean, it seems like the vipers get it pretty bad in the face as well, especially because it seems like they also get it around their nasolabial pits a lot, which is something I've wondered. Is it just because it's like a warm, humid opening in their face, which might be conducive to the fungal growth? But yeah, I remember from the inoculation trial that they did with cottonmouths, I think the only thing that they were able to show increased over time in terms of clinical signs was facial swelling. But they didn't necessarily see an increase in some of the other clinical signs that they were looking to measure. Yeah.
It's just bizarre.
Yeah, it's super interesting. It makes you wonder if it is spread through, specifically in the cases of the bad face ones, if it's spread because the animal is, I don't want to say excavating with its muzzle or touching its body with its muzzle,
going face first and everything.
Yeah. I mean, but I just think of kind of like how dogs always get injured on their nose because of that, because they're always putting their face into stuff, I don't know.
Yeah, that's an interesting point.
Has it led to anything like stomatitis or other sort of secondary issues like mouth rot or anything internally, like in the mouth itself? Have they done any studies on that?
I think when they've done some necropsies in inoculation trials, they've shown that there have been pockets of bacterial growth associated with the lesions. So like if you take a lesion, there's actual bacterial growth associated with it. Now, should I talk about some of the results that are associated with our statewide data set, or should I kind of?
Sure. Yeah. Go for it. Mm-hmm.
Yeah, and so when I was at a conference recently, one of the things that people were talking a lot about is co-infection in other reptile diseases, so like chytrid and things like that, which got me thinking about whether snake fungal disease might have co-infecting pathogens. And so I recently ran an analysis on all of our point captures from snakes across Tennessee, and basically found that snakes they had higher Staphylococcus in their microbiome than snakes that didn't. And if you look at snakes, as their amount of snake fungal disease increases in terms of like the amount of DNA that we measure, we're more likely to record the snake having Staphylococcus in their microbiome than not. And that was also the case with the bacteria known as Nocardia, which can also co-infect. So, for example, with white nose syndrome in bats. So those are two potential like co-infecting bacteria that may be occurring in the snake fungal disease system.
So yeah, you've got this animal that's got an atrocious, skin fungus, and then you're adding the compromised immune system and staph. It's screwed.
Yeah,
to say the least. Yeah.
Bad day.
Oh, poor guys, man.
But that staph is in those same lesions, like those same infected areas, those same pockets, or is that would be systematic?
We can't say that. All we can say, so this is based on analysis of microbiome data. So all we can say is we measure the DNA of the microbes that were there, and that this is the case with kind of the DNA markers that we measured. And then that was a separate study where they showed that there were bacterial lesions. And so I wouldn't venture so far as to say like those lesions are composed exclusively of staph or anything like that. That would be interesting for someone to follow up and kind of look at the composition of those lesions through time. But I do think that those results suggest that the development of disease is at least facilitating the kind of increase or enrichment of Staphylococcus in the snake microbiome.
And at this point, it's correlative.
Yeah.
Correlative, but an interesting trend, totally worth pursuing in the future.
Mm-hmm.
Absolutely. Super interesting.
And that's something just in general that I'm a firm believer in learning about like snake systems and how they work, like anatomy and things that you would think would be, I guess, fairly common practice with a lot of other groups of animals and people that keep them. But like the immune system, like I feel like there's so much about snakes and reptiles in general when it comes to that kind of stuff that I don't know that I really want to know, but I don't know that there's a ton of information out there as far as like how an immune system in a reptile operates differently from mammals or other things like that, because that just makes me wonder, like if they're getting those infections, what's their blood looking like? Is there a drop in oxygen levels there or anything like that? I'm just thinking out loud, but like that's the kind of stuff that I'm really interested in, is like what does it do to the entire system, not just the skin and everything else that's going on. But I have the Mater's book, and that's about as good of information you can get, and it comes to anything veterinary or medical with herps. So I don't know, I'm just constantly trying to find that information and learn about it, because I'm super curious, because I feel like I should know how that stuff works too. Like if you're keeping these things and you're dealing with them, like you should have a fairly, even if it's just a basic grasp of how they work internally, how their liver and kidney functions differ from other animals and why they work differently and what they, that's, I don't know. Just
agreed, man. Agreed. Yeah. Let me ask you this, since we're talking about a lot of live bearing specimens that you guys have worked with, whether it be, Neurodia or Echistrodon, whatever, have you noticed it transmit to offspring?
Yeah, that's a great question. We haven't like taken an animal into the lab and had to give birth or something like that, but there has been a paper about some tentative instances of vertical transmission. I think they give 3 examples, and to be honest with you, I don't remember all 3 of them off the top of my head. But I remember at least one was, I think, a king snake where they brought it into captivity, or one was a king snake and eggs actually. Where they thought that there was potentially some growth on the eggs of from snake fungal disease, and then I think some of the offspring they found qPCR positives on.
Wow.
Yeah.
The sample sizes were very, very small. It was just a few animals, but I think antidotal, something worth following up on, definitely.
Sure.
Yeah, it was essentially 3 like reports. Like these are 3 potential instances of vertical transmission, rather than like a study where here's like we generated or like we are documenting vertical transmission or generated it or something like that.
Yeah,
I mean,
you'd almost have to. What we're gonna say, Smitty?
Hypothetically, you would think with all that albumin and stuff coming out of the egg that it would provide some sort of a buffer, like a net, so to speak, to where it just traps those spores and they have a harder time sort of connecting to things. And
yeah, or it's the perfect conduit,
Yeah, that's it.
You could just be smearing it around, so to speak. Mm-hmm. I think it'd be very interesting if you had something like a gravid cottonmouth, and let's say she's hours away from dropping her litter. If you were to put her in a fairly sterile environment, and then the minute the baby comes out, you scoop up that little sucker while it's still in the amniotic sac and separate it, and then leave some babies in to be communal with mom, I imagine that you would get very unique results, regardless of what they were.
Well, that makes me wonder how much it hangs out around the vent too, because I mean, you think about a lot of the other parasites and bacteria and things. The fecal oral route is pretty common mode of transmission for a lot of stuff. So I wonder if it's if the snake fungal disease is any different in that aspect, to where it's like, yeah, this is the vent. This is where things get spread the most. This is where I should set up camp and hitchhike to be put somewhere else.
Yeah.
I just want to clarify. So that study on that vertical transmission paper, the Lampropeltis, so the king snake did lay eggs and did have growth on the eggs. That was correct. They're just none of them hatched because of the growth. I just wanted to double check that. And that is a paper by Stengel et al. From 2019. In case people want to double check that, just since I didn't have full knowledge of that paper.
Very interesting. And it was because of actual snake fungal. It was because of snake fungal disease fungus. It wasn't just that the eggs got moldy.
Yeah, I have it pulled up right here.
Excellent.
So swabs from 9 of the 10 eggshells were PCR positive for the fungus, and viable OO was recovered from one of the eggs.
Wow. That's even more scary. Wow.
Well, microbes are known to be transmitted vertically.
Yeah.
Right. Yeah.
Yeah.
Mother to offspring. I think snakes are no exception.
Yeah. Absolutely wild. I had a question. Now I forgot it. Damn, I was waiting for Doc to come back.
I had to grab a beer.
You had to.
and tuck the kid in. Yeah.
Oh, you got to do what you got to do. Fatherly duties, man.
Yeah.
I remember now. So over the past, say, 2-3 years, there's been many advancements, mostly thanks to our good friend Dr. Zach Loafman from West Liberty University, on Cryptosporidium transmission and prevention. And one thing is phorid flies. And has there been any documented cases of phorid flies cross-contaminating?
Being vectors.
Yeah, being vectors.
That
I wouldn't doubt it for a second, though, honestly.
Yeah, I imagine it would be super easy.
Yeah. I haven't read that work, but the fungus is known to grow on insect exoskeletons and fish scales and a variety of other substrates. But I'm actually not familiar. What type of flies are they?
Phorid flies.
Yeah.
They're very, very similar to fruit flies, but they're much bigger. They're less prone to actually fly. They're more of a walking fly, if that makes sense. And
they prefer dead stuff over. They're not like a fruit. Like they grow on decaying.
Yeah. Most people get phorid flies, first of all, mostly in Florida and the South, but they inhabit your drains. And the elbow in the drain, it gets all that that human funk in the elbow of the drain, and the flies live in there. Well, a lot of people, if you Drano your drains or you try and keep your bathroom clean, you really don't ever see them. Most people don't even know they exist. They may see one in their kitchen, in summertime, and they think, oh, it's a fruit fly. I left the apple out or something. When in actuality, it's not. It's the forward fly. Well, in snakes, we see them all too often because of snake poo, but mostly it's either regurgitated prey items or a prey item that was not eaten, or the animal killed it and left it, and then you forgot about it because you're not paying attention to everything, and all of a sudden it's been 24 hours and now it's starting to smell a little ripe, and then boom, now you get these little flies walking around everywhere.
And they go from cage to cage, spreading.
And they go from cage to cage. They go from room to room, spreading party favors. Yeah, protozoan for the full family.
Major vector for crypto.
Yeah, I think y'all are going to leave us with a number of ideas to follow up on in the laboratory.
Oh man, we try to have some
Armageddon. We're not going to send up actual astronauts. We're just going to get the oil worker guys to go up there and do it.
Exactly. Well, I tell you, man, I had forward flies this past winter, and it was brutal, and it wasn't even from the snakes, it was the drain, and I found out that they're doing construction in my neighborhood on the other side of the neighborhood, and they were like purging sewer lines and like putting in fire hydrants, and just somehow they got up in there, and it took me a good month of like diligent drain cleaning to get rid of those little suckers.
I am convinced those little electronic catchies that I bought for my room are like the number one preventative measure for those little bastards, because
I believe it.
I won't even see flies in my room, like fruit flies, anything. I won't see any, but I put that catchy in there. Those pads are covered in like 2 days. But I don't ever see flies. It's bizarre. They just show up.
Using the ribbons?
No, so it's this little unit that you plug into your wall. It's got a little fan and it's got a UV light on it to attract them, and then it basically sucks them in, and there's a sticky pad that goes in the very bottom of it, and it just shoots them onto that pad, and it just collects them like crazy. But it works like $1 million, man. It's like I bought 2 of them. I got 2 in my room, and I'll run them pretty much 24/7 and just change out the pads periodically. But I guess that might be a good method to collect some bodies to test.
Yeah, absolutely.
More than enough on there.
But yeah, I imagine that forward flies could really do a hurting if there was some kind of fungal disease in a collection, or and that's the problem too, is someone like myself, that I have multiple rooms for quarantine, right? And I got to make sure that if I see flies in one room, like I got to get the fly strip and start cleaning stuff out, and I have different tools for different rooms because I don't want cross contamination, because that little bugger, he could go in one drain, come out your air vent on the other side of the house, and you'd never know. And it's scary. I mean, I don't want to live in fear. I don't want other people to live in fear, but yeah, I imagine it could probably put a hurt on something.
Yeah.
Well, that got morbid quick. Sorry, guys. Just the cogs are turning,
Yeah, totally.
So what's the next step for you guys in this regard?
Yeah. Well, Alex has different next steps than I do. Alex is going to go on and pursue a PhD in another laboratory. He's going to turn out to be he's already a fantastic scientist, but he's going to go on and lead a laboratory elsewhere at some point, and maybe we'll continue to study snake fungal disease together in the future. But for my laboratory, we are actively recruiting students at the master's, PhD, and postdoctoral level to study snake fungal disease. We're interested in linking field studies to lab-based live animal studies to what we call like synthetic microbiome studies. So what we're doing now most recently is using these really cool controlled experiments that we can run in 96 well plates, where we can attempt to mimic like the composition of the snake skin using like keratin, fat, variety of different fatty acids and lipids. We can try to mimic snake skin in a liquid medium, and then we can introduce Ophidiomyces to a variety of different bacteria and see how they interact on this medium that mimics the snake skin. So we're really trying to, so we're really trying to kind of link what we see, the patterns that we see at broad scales in the field, to live animal studies in the laboratory, all the way down to these really tightly controlled laboratory experiments in Petri plates, more or less, or 96 well plates, just to improve the ability of replication, just to understand how bacteria and fungi interact in these simplified experiments.
Very interesting. Very, very interesting. It's awesome.
Yeah, we're interested in it from like a theoretical standpoint. We're interested in it from, fundamental science, perhaps learning how different bacteria may suppress the fungus from growing, a variety, which is more applied if you're thinking about like bioaugmentation, like changing the snakeskin microbiome to attempt to fight off Ophidiomyces, put it very generally, for different like breeding and reintroduction type programs. But yeah, we're interested in it from theoretical, fundamental to applied sciences, just understanding how bacteria and fungi interact on animal skins and also inside of the gut of different reptiles and amphibians.
Wow.
Are there microbes that eat the spores, like predatory type?
Oh, I'm sure there are. Yeah. Mm-hmm. Yeah, I'm sure there's probably a variety of different protists that eat spores. I'm sure that there are, or not, I'm sure there are definitely fungi that parasitize other fungi. I'm sure bacteria probably live and grow on the fungus as well. When it's no longer living and growing on the snake skin, right, as it grows and kind of grows across that leading edge and leaves behind dead and decaying cells. I can imagine that bacteria probably live and grow on the fungus itself. I think these are all interesting questions that could be investigated.
Yeah, so if either of you guys want to do a PhD in the Walker Lab, just send dummies.
Yeah.
Alex, I mean, how much of a concern do you think it should be for private collections? I mean, obviously, if you're buying wild-caught snakes from like native U.S. stuff, especially, you kind of have a higher chance of getting it. But I mean, in general, to me, it doesn't seem like it's something to worry about as much on the level of like crypto and other major, serious outbreaks of possible pathogens. But what's your take on that?
I agree with that. I think in general, it's not the most contagious pathogen you could see in the reptile trade, which is fortunate. But I do think that your treatment options are fairly limited if you do have it in your collection. Like, so because I work with snake fungal disease so much, like whether I was having it in, running experiments with it or just working with it in the field, like I just have a corn snake right now. Like it's not the, the most exciting animal collection, but I've always been very concerned, like, the last thing I want to do one day is come home and see my snake's face swollen and think like, oh my God, like, am I going to have to like bring him to a vet and try to convince him like, listen, you need to nebulize him with terbinafine. Like, I know you don't know what this is, but I do, you know what I'm saying?
Yeah.
So I think it's definitely a concern for people who, and, North American snakes definitely have it, but it is also in Europe now. It's in Asia. It's really unknown whether it could be in Australia or South America. So I think in general, people who are buying imports need to generally be on their toes about it. It does spread from animal to animal. So even though it's not necessarily the most contagious disease, if it gets into your collection, I think it could be a really bad thing, particularly because treatment options are, it takes long-term treatment and it's not an easy thing to treat. And yeah, I think it should definitely be on people's radar. And then if for no other reason, then even if it's not necessarily the most pressing concern for captive collections, a lot of people who keep reptiles like to field herp. They're appreciative of wild herps. And it's definitely something that's present in wild populations.
Yeah, that would be the biggest thing too, aside from imported animals or wild caught stuff, is like if you're out herping and whatnot, making sure that you're stripping down and getting clean before you go into your room, or not even go in your room at all.
Yeah.
Like I could see that being a much more plausible happening than an import necessarily. Like I can, especially I guess it depends on how much herping you do. You know, if you're out and about a lot and you're walking around and stuff, and then you decide to just walk into your room and clean some stuff or whatever, then you're one giant vector yourself.
Yeah. Or I mean, in the case of myself, I mean, there was a good time where I was trying to, you know, work with as many Florida native stuff as I could, and I wound up keeping several animals, and I would literally bring it right home. And unfortunately, because of Florida laws, I'm not allowed to, but during my current situation, I cannot have a venomous quarantine. So aside from it being on the other side of the room in its own isolated enclosures, when I take a pygmy rattlesnake home, it's in the same room, you know? So obviously if there's something visually wrong with the animal, I won't keep it. But, you know, how do I know it's not there, you know, dormant or whatever? So it's a good point. Very good point.
Especially stuff with crazy, you know, a lot more heavy patterning and contrast and stuff, where maybe seeing some of those skin or abnormalities, especially something you're not hands-on with, it might be harder to catch.
Yeah.
Especially when they're so small, like pygmies are. Damn.
Yeah. Yeah, and they're so keeled, and such a dynamic pattern on such a keeled animal that, you know, and not to mention we always joke about jungle skin. Like it takes at least 4 or 5 sheds before that animal looks like it's supposed to. You know what I mean? Mm-hmm. So just need to be observant.
Those Fiji bottles come in handy. You can get that magnifying glass out, buddy.
Dude, I've done it. I do it every time. Every time. So
Fiji bottles are the pygmy rattler catch container of choice for Phil there.
It's a long story. We could talk about it off air. So, but yes, Fiji should endorse this show.
And bang.
and bang, yes. So, well, Smithy, is there any other questions you want to touch base on?
I don't think so. I mean, Harry asked, you know, about predatory mites having any interest in fungi. And then Billy Jenkins mentioned maybe isopods eating spores or springtails. Springtails, I can definitely see.
Yeah.
Being a part of that. And isopods too, I'm sure, but springtails being as small as they are and the numbers they're in, depending on how well you have them built up in an enclosure, you know, I can definitely see those spreading it like crazy.
And I also imagine it would be more of a preventative, but it's not going to cure anything, you know?
Mm-hmm.
Yeah, that would definitely be my thought.
Yeah.
The only way I see predatory mites having any interest in it is they're eating a mite that has it on it.
That's true. Henry also messaged me asking, to assume that this was not done in study, but he wanted to know the effects of the degradation from cytotoxins as well as hemotoxins and myotoxins in an animal that has contracted the disease. So if he's feeding it to an elapid that has predominantly, you know, cyto and myotoxic venom, is the degradation in the GI tract going to essentially kill the fungus? Or is it kind of a moot topic because the snake already had its mouth all over it and it's, you know, it's there?
That would be my thought. I would assume that it's the process of actually consuming the organism is probably where it's going to be picking up that infection. And that was actually, I meant to mention that before when we were talking about like concerns for people like in husbandry. People who do keep Ophiophagus snakes would probably be another one of those people where it should really be on the radar.
Yeah. I mean, it's kind of like eating hot wings. You're gonna get sauce in your face. You know, gotta make it jovial. Sorry, guys. Gotta make it not so dark. I mean, we wanted you guys to come on, and I feel like it's just getting morbid as hell.
No, I mean it's interesting because I mean it's something that you gotta. It's a very real consideration for especially for people that are out in the field a lot.
Yeah. Oh yeah.
But
well, gentlemen,
I don't have anything else. I don't know if you do, but
no, I mean, I feel like I went through the little Rolodex of questions I had in my brain. Yeah. I guess you guys want to pick who goes first. Where can people find you guys?
And what's the plan moving forward too, in terms of new information coming out?
Oh yeah, that's a good one.
Yeah. So we definitely have one paper coming out, hopefully early next year. That is going to be our spatial data set. So the last paper that you saw me post up on my Instagram about was my master's thesis, and that was like an inoculation-based experiment. So this paper is going to be, we looked at a bunch of snakes across time. It's like 800 snakes over about 5 years. And so that's kind of looking at like, did we find the same trends that we found in the landscape, in the like live animal experiment on the landscape? And so far, we're finding a lot of very similar trends, which is great because that means that, you know, I didn't spend 2 years on my master's investigating nothing.
It's good, man. That's real good.
Yeah, for sure. Believe me, you're telling me. And then we're also, I'm working on that shedding data set, you know, so those are two things that we're plugging away on. And then we're also working on the Basidiobla stuff that Donnie, or that we talked about before. So we have some other stuff coming out about herps if people are interested in kind of just generally that interface of microbiology and herpetology.
Oh yeah.
Yeah.
You
there,
can
like a single channel where people can stay up to date with this stuff? Like you guys have like a Facebook page or a website that.
So if people are interested, my Instagram these days is asromer_biology. So I do post if I like publish paper or something like that on there, and then I also post a little bit of like, you know, when I'm out in the field doing photography and that kind of thing. And then I have a research gate where people can just find me with the name that I have here, Alexander S. Romer, if people are interested in following me more on the academic side of things. And then Donnie, we do have a lab website, right?
Yeah, we have a lab website. Yeah, if you look up the Microbiome Ecology Lab at Middle Tennessee State University, you should find us, or just Google my name, Donald Walker. I know I'm on the screen as Donnie, but my name is Donald Walker. If you Google me, you'll find, and in the word microbe or fungi or whatever, you'll find a bunch of different resources. I have a Twitter account. I go by DMW Microbiome. So awesome. I do post a little bit there, but not a whole lot.
Yeah, we could definitely get the new papers, and we'll throw them up on the network Facebook page and the website
awesome.
and stuff.
Anytime
Much
you guys got new
appreciated.
information that needs to get out there, let us know, because we'll
Much appreciated.
gladly throw it up there. So
thank you.
Yeah, we're trying to get as much exposure as possible. It's important to promote an understanding about all of this.
Hell yeah. Absolutely awesome.
Yeah, well, this was an awesome show. I'm really glad we made it happen.
It really was.
Yeah.
I'm glad you guys enjoyed it.
Yeah, thank you guys for inviting us. Really appreciate it.
Yeah. Hell yeah.
This episode was brought to you by BlackBoxCages.com. Check them out. Facebook, Instagram, BlackBoxCages.com. And Puget Sound Pythons. Check them out. Facebook and Instagram. They, like I said at the beginning of the show, should have some stuff available here soon. I know they hatched out a handful of ball pythons, and they got some other odds and ends. So hit them up, see what they've got. I think I don't know if we're doing THP this coming week or not. I know Chris and I are doing a Corn Stars episode Wednesday. So that is to be determined. But CS is definitely happening. Siesta.
Sweet.
THP up in the air. So
rock and roll.
We'll see. Thank you, everybody.
Yes.
Yeah.
Thank you, guys. And next time Dr. Donnie will be on, he will have some facial hair. Life is going to be good.
It won't happen.
Well, hey,
thanks.
guys, thanks so much, guys.
Thank you.
Thank you.
Thank you.